My name is Yuki Fujimori. I'm going to present my research project. Green tea polyphenol epigallocatechin gallate modulate TNF-alpha secretion and CCR2 gene expression on human THP1 macrophages. Green tea has been reported to have a variety of health benefits. Many of these beneficial effects of green tea are related to its catechins, particularly EGCG as content. The present study focused on the effect of EGCG as a main component of green tea extract on the human immune system using human monocyte THP1 cells. Differentiated THP1 cells are treated with EGCG for the present study. The amount of TNF-alpha was measured using ELISA, and CCR2 gene expression after EGCG treatment was determined by real-time qPCR. It was hypothesized that EGCG treatment would reduce TNF-alpha release by a dose-dependent manner and would alter CCR2 gene expression following TNF-alpha release levels. EGCG extraction was performed by brewing green tea, extraction of catechins, and decaffeination. The cells were treated with EGCG in two different ways: co-treatment, EGCG and LPS at the same time; pre plus co-treatment, EGCG first, then add LPS without removing EGCG treatment. The cell viability of THP1 cells in EGCG treatment was evaluated by MTT assay. TNF-alpha release in each sample was detected by ELISA. CCR2 gene expression was detected by RT-qPCR. Data analysis was performed by following. These chromatograms show the components in green tea extract after each extraction step. Since there is a detection limit on UPLC system, very high concentration of EGCG couldn't be detected at all, so I did tenfold dilution. Other peaks on the chromatogram were other catechins in green tea extract. 50 microgram per milliliter of EGCG treatment showed significantly lower cell viability. However, other EGCG concentrations were not significantly effective on THP1 cell viability. The left side shows the result of co-treatment, and the right side shows pre plus co-treatment. At 6 hours, both treatments didn't show much TNF-alpha release on every concentration. However, at 24 hours, co-treatment showed significantly higher amount on 10 microgram per milliliter EGCG treatment and significantly lower amount on 500. On the other hand, pre plus co-treatment showed the lower amount of TNF-alpha release on 10 microgram per milliliter than control, and 50, 100, and 500 microgram per milliliter showed significantly lower values. Since some of CQ values from PCR testing showed invariant numbers, it wasn't able to be taken for Turkey's comparison test. However, the CCR2 gene expression showed the similar manner as TNF-alpha release level on co-treatment. The results showed EGCG was more effective by treating cells before adding LPS treatment. The present study provided a glimpse of a potential inhibitory effect of pre-plus co-treatment on TNF-alpha secretion. The real-time qPCR result also showed that EGCG might not simply downregulate TNF-alpha secretion, but it might reduce it by inhibiting CCL2 to bind to its receptor CCR2. However, the data collection of this study might be able to be improved with a higher seeding density of THP1 cells, and the contribution of EGCG2 immune response could have been clearer by performing real-time qPCR on pre-plus co-treatment samples.